So next we have the cytoskeleton of the body. Cytoskeleton means cyto means cells and skeleton means the cellular skeleton. So the cellular skeleton of the cell is actually the microtubules, intermediate filaments, and the keratin, the vimentin. They all are the cytoskeleton. So in the cytoskeleton, the most important one is the microtubule. Microtubule is M-phase specific cytoskeleton, and it is used in cellular division, making the spindle fiber, making centrioles, making cilia, flagella, doing axonal trafficking. So these all functions are related to microtubules. So microtubule, the structure of microtubule, they're actually a cylindrical outer structure which is made up of a helical array of polymerized heterodimers. Heterodimers are actually the tubulin, alpha and beta heterodimer tubulin. So alpha and beta heterodimer tubulin, they actually bound to 2-GTP bind to these tubulins. They get polymerized, they attach one by another, make a helical array, and this helical array make a proper cylindrical shape, and this cylindrical thing is called microtubule. So 2-GTP are attached. So there are two ends of microtubule, the negative end or a positive end. Negative end is near the nucleus and positive end is pointing the periphery. So there is a protein known as tau protein, which is very much important when we are studying neurofibrillary tangles in the Alzheimer's disease. So tau protein actually stabilize the whole structure of microtubule. So if tau protein is abnormal, like in Alzheimer's disease, the tau protein is hyperphosphorylated, which is no more stabilized the microtubule, then the microtubule damage. So no stabilization of the microtubule, it gets collapsed. So this is the mechanism or the relation of CNS pathology with this microtubule. Also microtubule, they are used in cell division and spindle, mitotic spindle. They also do slow type of exoplasmic transport using the two molecular motor proteins. These two molecular motor proteins are actually the dynein and kinesin. Dynein are actually the retrograde type of protein, and kinesin is actually anterograde type of protein. Dynein move the substances from positive end, or from positive or periphery to the nucleus, or negative end, periphery to the nucleus. And whereas kinesin, they move from nucleus negative end to positive end, periphery. In this way, dynein is important in the latency of different viruses like polio virus, like rabies virus, like herpes simplex virus, and also herpes simplex, polio, and tetanus toxin. They use pretty much dynein in order to get latency. But whenever they want to activate, for example, this herpes type of lesions, then they use kinesin. So kinesin is for reactivation. After this, we have the drugs which inhibit the microtubules. They are, first one are the anti-cancerous drug, the taxane family. Taxane family like Paclitaxel or docetaxel, they actually hyperstabilize, like uh, they actually, taxane actually hyper stabilize the microtubules, so there is no cell division. So no cell division, no centriole formation, no mitotic spindle formation. So now the cell is going to die because there is no replication. So in this way, the taxane family control the tumor cells, and the side effect is pretty much important, that is they are neurotoxic, or they can cause neuropathies. So this is the important, the most high yield side effect of taxane family is they are neurotoxic. The next family is the vinca alkaloids, and vinca alkaloid we have the vincristine and vinblastine. They don't follow the hyperstabilization, rather they bind and inhibit the tubulin to undergo polymerization process, and in this way they inhibit the microtubule structure formation and inhibit the cell division of cell, in this way inhibit the cell division. So vincristine, they actually, the side effect is important, they crisp the nerve, so they are neurotoxic. And vinblastine, they, in addition to crisping, they can cause blasting the marrow, causing bone marrow suppression. After this we have colchicine used in gout. We have mebendazole and grazofulvin. Mebendazole used in protozoal control, and grazofulvin is an antifungal agent. So these are all about the microtubule structure. The next structure regarding the microtubule is cilia, and it is pretty much straightforward. Cilia is actually a 9 doublet and one singlet, or two singlets structure. 2 singlets in the center and 9 doublets are in the periphery. They make a cilia structure. So the basal body of the cilia, they have 9 triplets, and there is a exonemal dynein which connect all the structure. So exonemal dynein, which is associated with the ATPase enzyme. So ATPase is linked with that dynein, and this ATPase use ATP molecule to generate a differential sliding or movement, and this movement is help the cilia to make ciliated mucociliary clearances type of function in the ciliated epithelium. So there is a gap junctions which make this whole movement pretty much coordinated, a rhythmic or coordinated fashion. And we also have a nexons which connect all these 9 tablets together. So this is the main structure of cilia. If this cilia is abnormal, then we have a condition known as Kartagener disease, which is a primary ciliary dyskinesia, primary ciliary dyskinesia, and it is actually an autosomal recessive condition in which there is a dynein defect. If this dynein is defective, then there is no motility of cilia. So cilia are immotile. So all the ciliated epithelium is dysfunctional. So these three are very much important. If the dysfunctional epithelium, if the ciliated epithelium is dysfunctional, then there is no mucoclearance of the of the pathogens. So there is recurrent infection because no mucociliary clearance. So the patient is suffering from sinusitis, bronchiectasis, bronchitis, a lot of different infections. If the cilia are immotile, then there is infertility in the fallopian tubes. There is dysfunctional cilia which can lead to ectopic pregnancy, or there is infertility in the males, like the immotile sperms. And this dynein defect is not only also there is a less common symptom, that is hearing loss, in which the organ of Corti, they use hairy ciliated cell to transmit. So there is a conductive type of hearing loss. So this dynein defect is not only just related to this cilia, but also it is important in the migration and orientation of different organelles during developmental phase. So there is there is a defect in dynein, so no migration, no orientation, so there is developmental abnormalities. And in this developmental abnormality, situs inversus is very much common. So if we have to screen the newborns by checking their nasal nitric oxide stimulation, so if there is a decreased nasal nitric oxide, then the patient is suffer from Kartagener disease, or it needs to be evaluated further. So this is all about the microtubules, cilia and microtubule and their related anti-cancer drugs.